Hi everyone, hoping that you all are well. Thank you for joining us for our discussion related to this new bat virus known as KHOSTA 2. This virus is supposed to be similar to the current situation. It's another microbe that is attracted to what's known as ACE2. We're going to also be looking at some of the symbology and mythology that's tied to this name choice. So we're going to be looking at some issues related to taxonomy and nomenclature. One thing to point out as we venture into looking at what some of the science has to say about this particular microbe is that this microbe is apparently resistant to antibodies related to the current situation, and that includes a resistance to the inoculations that have been received over the last year or so, and that includes the most recent iteration of those inoculations. In terms of taxonomy, as you can see, this particular microbe is identified as bat SARS-like, and it's referred to as KHOSTA 2. There's also a KHOSTA 1. This KHOSTA 2 microbe is reported as being found in a bat out of Russia. And similar to the current situation, it also contains a spike glycoprotein. It's also interesting to note that the science literature indicates that the glycoprotein is also something that is related to the venom that bats have. Now, even though this bat is a Russian bat, the primary research is coming from some American researchers. We're just going to look very quickly at a couple of research articles to get an idea of some of the science data and some of the scientific concerns that have been articulated. This particular article, this comes from September 22nd, 2022, and it indicates an ACE2 dependent SARS-CoV-2 in Russian bats is resistant to the current inoculations. And the lead researcher is indicated as focusing on decoding the rules of host virus compatibility. It is pointed out that while there are similarities between the current situation and this KHOSTA 2, there are some distinctions. It's also indicated, however, that this particular KHOSTA 2 utilizes the ACE2 in order to gain entry into the host cell. Now, in this instance, we are talking about the animal that becomes infected, and specifically, we are talking about humans. There's also an indication here that this particular microbe is resistant to monoclonal antibodies and serum from individuals who have been inoculated with respect to the current situation. The researchers also indicate that there are hundreds of these SARS-CoV viruses that have been discovered, and they are primarily in bats in Asia. However, the majority of them are not capable of infecting humans. What has transpired, however, with the KHOSTA 2 that was discovered in Russia is that with some manipulation, this particular microbe is now able to infect human cells under different conditions. The researchers go on to make a notation about many animal species carrying their own microbes. They also indicate that with the discovery of additional ACE2 dependent SARS-CoV viruses in broader geographic regions, the risk of new recombinant viruses is rising. It's noteworthy that many of these individuals, they don't seem to take into consideration the possibility of these things not spreading if they would simply leave them alone. And so good fences make good neighbors, and that might apply to the animal kingdom also. Now, the researchers go on to point out that there is similarity between the current situation and this KHOSTA 2, but they also point out that this particular microbe, this KHOSTA 2, is least similar to the currently circulating situation, and that this also has implications for this resistance to these inoculations. These researchers go on to assert a belief that new inoculations are needed to address the resistance that this KHOSTA 2 has to the inoculations that are currently in use. This Science Alert article provides additional reflection related to this KHOSTA 2 microbe. And here they note some of the similarities between the current situation and this KHOSTA 2. And what they indicate is that both are respiratory viruses. This virus, however, is known as KHOSTA 2, and it is covered in spike proteins that can infect human cells using the same airways. They also point out that this new respiratory virus cannot be neutralized by current medicines that are in use. This article offers that some of the traits of this KHOSTA 2 are worrisome. What they indicate is that the bat pathogen was able to use the ACE2 receptors on human liver cells to infect tissue in the same way that the current situation is able to do. This article specifies that this particular species, this bat species, is also found in Europe and North Africa. Additionally, what is indicated here is that they are not clear if this particular microbe that infects these bats can spill over to humans in the real world. And so they can see it in the lab, but they are not clear that it can actually happen in what's called in vivo. But their findings suggest that this is certainly possible. Another concern that is articulated here is that if someone comes down with the KHOSTA 2 virus and they already have the virus for the current situation, that these two viruses may actually recombine in order to create an entirely new variant. The article goes on to reiterate a belief that new strategies need to be put in place to address the fact that this particular microbe is not neutralized by the current inoculations and it is not neutralized by the current antibodies. They also point out a belief that there needs to be broad protection against these entities known as SARS-CoV-viruses. Notably, the Weather Channel is also commenting on this new microbe, KHOSTA 2. What they are indicating in this article is basically reiterating what has been noted in the other articles. Experiments were carried out to establish whether or not the current inoculations would be efficient with respect to the KHOSTA 2. What they found is that, according to the study, none of the human antibodies created by the inoculation could neutralize this KHOSTA 2, and so it's highly resistant. This KHOSTA 2 also appeared to be resistant to both inoculations that are currently offered in the United States. They also indicated that they tested the serum from two persons who had the current situation, a variation of the current situation, but the antibodies in them proved to be completely ineffective.